Okay, good afternoon, everyone. Welcome to our first event in this year's Provost Distinguished Speaker Series for our academic year. This series is an annual event where we invite our recently appointed Board of Trustees, distinguished professors, and endowed chairs to share their scholarship with us. I am Carl Loew. I'm a professor in psychological sciences and the provost at the university. We're really pleased to share this opportunity today to bring our community together in appreciation and celebration of the exceptional work we do at UConn. I would be remiss if I did not acknowledge the work it takes to arrange this series, which has been managed primarily by Amanda Pitts in our office in partnership with, I was going to say new, but he's been here for a year now, Vice Provost Michael Bradford, who you'll hear from in a little bit. I want to particularly thank Professor Leslie Loew for sharing his scholarship with us today. First, we've asked the School of Medicine Dean, Bruce Liang, to introduce Professor Loew and his talk, The Inner Space of Cells. But before I do that, I did just want to say a word about Dean Liang. He's an internationally renowned cardiologist and researcher. He's made important contributions to signaling mechanisms that regulate cardiovascular functions using both cellular and pharmacological approaches. He's been at UConn for almost 20 years now. He came as the chief of the Division of Cardiology and as the Ray Niag Distinguished Professor of Cardiovascular Biology and Medicine. He's held several leadership roles at UConn Health, including serving as the interim dean in 2011 and 2015, and as the director of the nationally recognized Pat and Jim Calhoun Center for Cardiology since 2003. But of course, we know him now as the dean of the School of Medicine. Having graduated from Harvard Medical School and a residency at UPenn, he serves as a great reminder of not just the amazing faculty researchers we have at this university, but also among our administrators. And Bruce is a tremendous example of an administrator who remains and has truly been an outstanding researcher, a great scholar, an amazing colleague, and a wonderful teacher. So I'll hand it over to Bruce so he can introduce our featured speaker today.
Well, thank you very much, Carl. I wasn't expecting that. It's very kind of you to say those things about me. But I'm about to introduce today's speaker, who is more impressive than I am. If I were to talk about Les Loew, I could spend the whole time. So I have to restrain my remarks to give the amount of time he deserves. And congratulations to you, Les, for winning the Board of Trustees professorship. And thank you, Carl, for shepherding through the process. We're all very proud of Les. So Les Loew is professor of cell biology and holds the Boehringer Ingelheim Chair in Cell Science since 2007. Les holds a BS degree from the City College of New York, a PhD from Cornell University, and did postdoctoral fellowship and research at Harvard, all in chemistry. He has made a number of seminal contributions in the area of science. I will, it'd just be too much for me to mention. I would just highlight two major scientific accomplishments. Dr. Loew pioneered the design and synthesis of fluorescent voltage-sensitive dyes as a main area of his research foci. And he also pioneered the development of virtual cell. It's a computational system for modeling and simulating complex biological processes. And he invented the virtual cell, which has now over 25,000 registered users worldwide. So it's not just the US. And he's published one book, 237 research papers, and holds 9 patents. Over the 36 years career at UConn School of Medicine, he was awarded over or close to $70 million of extramural grants. With his two colleagues, also from UConn, he founded a company called Potentiometric Probes. And many of us aspire to such entrepreneurship, and we struggled for a long time, and maybe we have a product at the end of it, and Les already has a product which is actually garnering commercial activities. So a real remarkable accomplishment. And I can go on for a long time, Les. I would just mention two other things. You established the Richard D. Berlin Center for Cell Analysis and Modeling in 1994. You stepped down from that position a year ago, handed over to a very capable leader as your successor, but you continue to be extremely productive, heading up a center grant from NIH, as well as running your company, and recently assumed a leadership position for the School of Medicine as liaison for faculty who are interested in entrepreneurship and commercialization. And you have also served as editor-in-chief of Biophysical Journal, that very well-respected and high-impact journal that many of us aspire to publish in. So without further ado, I'm going to hand the podium over to you, Les. Thank you so much.
Thank you so much, Bruce. And thank you to all the organizers and to the Provost for this special honor. So I've sort of distracted you by having my slide up all this time, my first slide, to give you a chance to read it, so I don't have to read it to you. But the point is that biology is very complicated, and I can really relate to the subject of this cartoon, because I almost feel like this is me teaching biology. I really get very easily befuddled by all of the different molecules that are involved in biological systems and how they interact with each other and feed back on each other. There are thousands of different states of these molecules that they have to go through, and the way they interact. So I could easily, my wife can also attest to the fact that I can't really keep track of or focus on more than a couple of things at a time. So I don't know how biologists do it. I was trained as a chemist, which is also pretty complicated, but not nearly as complex and far-reaching in terms of what you have to know as biology. So, but on the other hand, it's also fun to contrast biology and physics. As many of you know, the holy grail in physics is sort of the opposite of all this detail. It's to develop a single equation that can explain everything. That's what physicists really aspire to. And there's an old story, which all physicists, I'm sure, in the audience may have heard. There once was a farmer whose cows suddenly started producing less milk. He had a long-term boyhood friend who became a world-renowned theoretical physicist and at a local university, and he asked him for help. The physicist took the challenge, and after 3 weeks of focused research on the subject of how to get the cows to produce more milk, here's what he came up with. So I'm sure you're all muted, but if you weren't, I would hear peals of laughter. But this is, I hope my physics colleagues aren't too offended or pained by this old joke. In seriousness, I have had many illuminating interactions with physics colleagues who are really able to simplify complex systems and truly see the forest for the trees. That's really what this joke is about. So I hope the physics people in the audience will forgive me. And I'm going to talk about physics concepts during the course of the next half hour or so. So here's the cover of a textbook that I admire on cell biology, and it's a beautiful cover. It looks like it's some sort of science fiction fantasy world, but it's actually an accurate depiction of many of the molecules and many of the structures that are inside cells. And you can see that a uniform density cow or a uniform density cell would not do it. We need to get into some of the details in order to understand how all of these individual objects work, as well as how they interact with each other. So the other aspect of this is that cells really aren't spherical. Here's a cell, it's a neuron called a Purkinje cell. It's a cell that's found in the cerebellum. There are about 15 million of these in the cerebellum. Each one of these cells, this is one cell, but each one of these cells gets 200,000 synaptic inputs from different cells. So the complexity is really mind-boggling. And there are about 80 billion neurons in the brain. They all have different shapes, different functions, different neurotransmitters that are involved in signaling to them. And so trying to understand even one of them is a great hurdle. And you can imagine how difficult it is to understand how they all interact. So you really need a multidisciplinary approach to be able to deal with this kind of complexity. And by the way, if we drill down still further and look at just one of these protrusions, which are called dendrites, and magnify them still further, you see that there's even more complexity at that level. So these little fuzzy areas, when you magnify them, turn out to show these cylindrical structures with these protrusions coming out called dendritic spines. And we'll talk more about that later. So again, just to give you an overview of what the complexity of a cell might be, in this case, a neuronal cell. So finally, I'm getting to my title slide, the inner space of cells. And I have a subtitle here, Multidisciplinary Studies of Cellular Systems. And that's really my main take-home message, is that you need to not just look at biology as biology, but look at biology as composed of physics and chemistry, complicated chemistry, and bring all of the tools that are available to you to try to understand it. And that's what our center, which Dr. Liang briefly mentioned, the Center for Cell Analysis and Modeling, is all about, to bring people together who can have backgrounds in math, physics, chemistry, like myself, computer science, engineering, biology, bring them all into one place and have them be able to talk to each other and try to solve problems in understanding cellular systems. And the kinds of approaches that we use and that we develop and that people outside our own center have ended up using are computational modeling approaches, live cell microscopy, quantitative live cell microscopy, and the development of fluorescent sensors. And I'll be telling you a little bit about some examples, some vignettes of how these projects have progressed over the last 10 or 15 years. So first, let's start with the virtual cell project, which again, Bruce mentioned. This is perhaps the biggest project in the center, and it's the biggest project that I've been involved in. It's been around now for 23 years. and has been continuously funded by NIH. And these are three of the people who started it. Starting from the right, Boris Levchenko is a physicist. He has his background really in solid state physics and mathematical physics. Jim Schaff is a UConn graduate, a genius engineer who was the main software developer for Virtual Cell. And Jan Muraru is pretty much not easily categorized. He's actually formally trained as a surgeon. He has an MD in surgery, but then he got more and more into research, did some cell biology research, but over the last 15 or so years, probably longer, he's, I think he now considers himself a computational biologist. And he's currently also the head of the high performance computing center here in CCamp. So he's also very knowledgeable in terms of computer hardware. So those are the three founders of Virtual Cell, but we've had people join along the way, and Ann Cowan is now the director of outreach and training. She's a cell biologist. And Michael Blinoff joined about 10 years ago, I think, maybe even a little longer by now. He's a mathematician and has added some really important new features to the software. So what is Virtual Cell? So this is the only equation that I'm going to show you, but I'm showing it to you because it really does show how you can write one equation, just like physicists love to do, and really describe a system as complicated as a cell. Of course, I'm not describing everything about cells with this one equation, but it actually does a pretty good job describing a lot of features of a cell and how it works. And the left side of the equation just says that the concentration of a particular molecule or state of a molecule inside the cell, molecule I, as a function of time and space, is given by the divergence of a flux on the right side. Now, by flux, I mean both diffusion, how molecules move around cells, and flow, how molecules might have a directed velocity inside a cell. So that's the first term on the right side. And the second term is sort of this R sub I is a grab bag term for all the ways that this molecule I can be created or consumed, or the state of molecule I can be created or consumed. And it, of course, can be a function of the states of other molecules in the system. So R sub I, although it looks like a simple term, is actually very, very complicated and can have a very large mathematical expression associated with it. But still, this one equation, which is really a system of equations, describes what Virtual Cell does pretty well. So there are currently 24,200 plus registered users for Virtual Cell. It has a graphical interface that makes it pretty straightforward for biologists with some physical chemistry background to be able to build models and do simulations. The kinds of mechanisms that are incorporated in this equation here are reaction kinetics, diffusion, advection, which is another term for flow, membrane transport, and even the electrical properties of cells, the electrophysiology of cells. It uses a lot of, it can use or produce very different kinds of simulations. And it's built on top of a database. So when a user builds a model, he can save the model to our database, share it with other users, share it with collaborators, and ultimately, when they publish a paper, the users can make the model public and share it with the world. Experiments can be incorporated into the model, experimental data, and we can fit experimental data. And it also allows you to simulate real microscopy experiments, like this one that's shown in the upper right. That little movie loop that's been playing here is an actual virtual cell simulation that's built off of real geometries from a confocal microscope. So with that very, very brief overview of what virtual cell is, I thought it'd be fun to just give you a couple of examples of systems that we've used virtual cell on in my lab, and at the same time show you some of the complexity, the beautiful complexity of cellular systems. So the first one I want to show you is this very interesting cell called a kidney podocyte. And there are actually 3 of them here in the 3 different colors, just so you can distinguish them. And the important thing to show you here is how, when you look on the inside of this structure as it passes through again, you'll see that the podocyte produces these finger-like protrusions that are actually called foot processes for some strange reason, but that's biology, that interdigitate. So it's as if you were taking your hand and interdigitating your fingers. So you can, I think you can see that as the 3D structure passes by. And this is work done by Sibeli Falkenberg. So here's her picture, so you know who actually did the work. She was a postdoc in the lab. This is her going away party. And the idea here is that here's another picture of this structure. This is now a scanning electron microscope structure showing these interdigitated foot processes. These are each filled with a polymer molecule called actin. Actin monomers can polymerize to produce actin filaments, and those filaments can bundle to give some structural integrity to these very fragile foot processes. By the way, the scale bar here is 1 micrometer. So the diameter of one of these foot processes is about 200 nanometers. Just to give you a scale for that, a human hair is about 70 micrometers in thickness. So this is less than somewhere on the order of 1/100th the thickness of a human hair. So if we can magnify this further and now show it as a cartoon, here's a cross section of 1 or actually 2 podocytes, and they're actually connected. These interdigitating fingers are connected by this bridge, this red bridge of protein molecules called nephrin. And nephrin actually forms the filtration barrier between a capillary down here and the lumen of the kidney out here. So it's the barrier that controls the filtration of urine from the blood, from the capillaries into the kidney lumen. And obviously, this is a very delicate structure. It needs to be controlled. And once it breaks down, it causes proteinuria. And can lead to failing kidneys. So maintaining this very fragile structure is critical, and how that's regulated is not really well understood, especially since there are pressure variations, etc., in the kidney that you would think might be very deleterious to this very fragile structure. If we drill down still further and magnify down to just what's going on the inside of the podocyte, we see that the nephrin molecule, these red guys, actually extend into the inside of the cell, the inside of these fingers, and they signal the actin cytoskeleton. So these are some molecules that are involved in signaling the actin cytoskeleton, these polymers that bundle to produce the rigid structure that fills these foot processes, these finger-like structures. So regulating the actin is critical. And here's what Sibeli found. She took these electron microscope images that we obtained from some collaborators in San Diego, and were also part of the paper that was published. And she analyzed the images and built a 3D model, shown here in J, of the entire cell. Now, she couldn't use the actual structure because it was just too complex and would take too much computer time to numerically simulate what's going on in such a complicated structure. She had to get some symmetry out of it, and so she analyzed the width of the podocytes, got the standard deviation of the widths and the lengths, and produced this 3D model over here, which I'll show you on the next slide. So here's that 3D model with the actin polymerization and bundling activity occurring inside it. And among many other things that she found in this work, the thing that I wanted to highlight for you was that in this relatively simple model, a spatially uniform increase in this bundling activity, the molecules that regulate bundling activity, produces very strange behavior. So here's time 0, where all the actin bundles are sort of uniform in these foot processes. You see they're all uniformly green, and there are hardly any bundles outside the foot processes. That's why they're down here at blue. So there's pretty much 0 bundles outside, but inside there's about 2.5. Okay. Once we increase the actin bundling activity, we're doing that uniformly everywhere. We find that the foot processes, these structures, do something very strange. Some of them decrease their bundling concentration, which would be sort of a surrogate for them actually collapsing and causing proteinuria. Here's just a blow up of this one area showing that some podocytes remain fine, but others lose their bundling activity. And if we wait a little while longer, this is 2,400 seconds instead of 1,200 seconds, we see that it's still different. And we see that some of them actually increase their bundling activity while others decrease. So this reason that I found this so exciting was this is sort of a hallmark of what's called chaotic behavior. Such unpredictable and variable behavior in a system is a hallmark of chaos. And this is, of course, something that's studied by physicists all the time. It doesn't mean that this is non-deterministic. It's a totally deterministic behavior. If you do the same thing again, if you start off with everything uniform and produce the same kind of stimulus by uniformly increasing actin bundling activity, the same exact pattern is produced. However, it's not something that you could have possibly predicted without doing the calculation or without actually doing the experiment, if you could do such an experiment. So it gives us some idea of how these fragile structures have to be super regulated in order to prevent this sort of chaotic behavior. Chaos is the same kind of thing that people talk about when they describe weather systems as being prone to the flutter of a butterfly wing on one side of the earth, producing, 3 years later, a typhoon on the other side of the earth. So that's the kind of physics that we're talking about, and it's a very well studied field. And here's a manifestation of it in a very different setting in biology. So now I want to turn back to that Purkinje cell. This is the Purkinje cell, a different image of it than the one I showed you before, down here in the lower right. And here's a reconstruction of an electron microscope set of images, a set of very thin slices from an electron microscope. And again, this is a very small area. This is probably about 15 micrometers across. And each of these protrusions, these dendritic spines that we call them, are about 1 micrometer in diameter. They're sort of mushroom type structures. And we built a 3D reaction diffusion model of this using virtual cell. Sherry Ann Brown was the graduate student. She was actually an MD PhD student who's now at a faculty position at Wisconsin Medical College. And she did this work as part of her thesis. And if we start playing the movie here, oops, let's see if I can get this movie to work. I have to get rid of the laser pointer for a minute, I think. Well, I can't get the movie to work, it seems. Well, the next slide pretty much shows the same thing, but not quite as dramatically. So I'll just go to the next slide. If you signal a single spine, if you impose a synaptic stimulus to a single one of these spines, and each of these spines, as I mentioned earlier, the Purkinje cell has 200,000 synaptic inputs. So each one of these spines corresponds to one of those synaptic inputs. And what we're able to show with these simulations is that each spine ends up being its own separate compartment. There is diffusion from the spine to the shaft of the dendrite, but it's retarded. And it means that the signal, in this case, a molecule called inositol trisphosphate, accumulates in the spine and can cause additional downstream effects before it gets diluted into the shaft of the dendrite. So this interplay of the biochemistry of producing this signal due to synaptic input and the speed of diffusion is just right so that the molecule in question accumulates inside this structure. And then eventually it goes down. But it's around long enough to set off downstream effects. Okay? And this is actually a good transition to the next sort of topic that I wanted to talk about. And that is, how do we measure some of these effects? So we have fluorescent indicators to measure calcium. Those weren't produced in my lab, but we also have fluorescent indicators to measure voltage, which is the primary signal, electrical signaling in neurons. And those were produced in my lab. And the next slide shows those, some of those. This is a whole set of these dyes, and if you want to stare at the structures, you can stare at them, but I'm not going to dwell on them. But just to show you that they're all different colors. And the reason that we have different colors is that for different experimental setups, we might want to use different colors or combinations of colors. And in particular, if we want to measure calcium and voltage at the same time, we have to use dyes that are calcium indicators and voltage indicators that have different color properties, different spectral properties, so that we can filter them out in the microscopy system that we're looking at. So here's an experiment that does this. This is a microscope image, a real microscope image of a dendrite in a living brain slice. So there's brain tissue all around it, but we only put the fluorescent dye in one cell. This was done by Corey Acker. In this cell, we can measure both calcium and voltage. So we've got a fluorescent dye for calcium at one set of spectral parameters, one set of colors, and another dye for voltage at a different set of colors. And here's the calcium signal. This is 10 milliseconds in this one spine. Again, this is only 10 micrometers. We can't use any other way of doing this. We have to use fluorescence. If we tried to use electrodes, they're just too big to be able to pierce an object as small as a single dendritic spine. So here's the calcium signal, and here's the voltage signal. And you can see that the voltage signal goes up and goes down, but the calcium signal sort of gets integrated. The voltage signal is the primary signal. It releases calcium, allows calcium to get into the spine, and then it goes down a little bit, but then it accumulates after the second action potential, after the second electrical pulse. So this is the kind of thing that we can learn from this combination of techniques. And you can see how it feeds back onto the computational modeling. And we were able to model this kind of effect very precisely with high fidelity in the virtual cell models. So finally, I'm almost out of time. I want to give a little time for questions. I want to talk about what happens between cells. Another electrical organ besides the brain is the heart, close to Dr. Liang's own heart. The heart has individual cells, cardiac cells, that are electrically excitable. That's why we can do an EKG on the heart. The EKG is, however, very low fidelity. You can have maybe 10 or 20 electrodes at most and get a very low fidelity map of the millions and millions of individual action potentials, individual electrical signals that are occurring in each cell of the heart. The beautiful thing about voltage sensitive dyes is that each pixel on your camera acts as if it's an individual electrodes. So if you have a camera with 1 million pixels, you can, and you magnify that image that the camera is recording, you can actually get down to the level where each pixel is measuring the electrical activity of a single cardiac cell. Of course, you have to be able to get at the heart with the fluorescence light. And to do that, back in 2019, we collaborated with Peter Lee and David Figueres Rama in Spain, where they were able to do open heart surgery on pigs. You see the little feet sticking out here? That's a pig undergoing open heart surgery, and they used these cameras that Peter Lee had designed. David is a cardiologist, a cardiac surgeon, and Peter is a bioengineer. And he designed these cameras to be able to do these experiments. And what you can see is this record is showing a typical cardiac action potential that he can measure from a single one of these pixels. But if you can measure one cardiac action potential, then you should be able to measure a cardiac action potential simultaneously from the entire heart. You're using a camera, so why not take a video? So that's what they did. And here's a video of the heart showing the pattern of sinus rhythm. This is a normal cardiac rhythm. And you can also see, of course, the heart pumping as it's doing this. And this is slowed down a bit. I think it's slowed down by about a factor of 5, but you can see the recording up here. The pig heart, I think, beats at about 700 milliseconds per beat, something like that. So maybe 1.5 beats per second, or 40 or so beats per minute. So this is slowed down, but you can see the wave of electrical activity starting up here and moving down to the lower right of this living heart in a living pig. So this is the kind of thing that you can do. And as I said, you can look at each pixel and get an individual action potential, but you can also see this pattern of activity. And this happens, of course, because the cardiac cells have electrical connections that allow this conduction to happen and allow it to be regulated carefully. But of course, what happens if things go wrong? So instead of having this beautiful wave, you can get this kind of behavior. You can see, first of all, that the heart is not beating anywhere near as effectively. It's not contracting as effectively as it was before. And the experimenters, Peter and David, put this heart into arrhythmia. And you can see these sort of spiral waves. And they're easier to see in this rendering of the same data. So this is real data. This is not a simulation. And that takes us back to the physics again, because this is a famous paper by the famous, actually he was more of a mathematician than he was a physicist, but he did a lot of very fundamental physics, Alan Turing, a British mathematician who tragically committed suicide shortly after this work was published. But he was an absolute genius, and he contributed to computing as well as to this particular area of mathematics called reaction diffusion mathematics. And it's really his legacy that we're still benefiting from today. So here's really the only figure in this seminal paper. It's called On the Chemical Basis of Morphogenesis, where he tried to explain, using reaction diffusion equations, the same equation that I showed you at the beginning for virtual cell, how patterns develop on in animals, how zebras have stripes, how flowers have their beautiful symmetrical patterns. And in this case, we're back to our cow again, how this dappled pattern could also result from reaction diffusion. And you know, just coincidentally, this is really the only example that he has in this paper that he published that set off this entire field, showing the dappled pattern that you might find on the hide of a cow. So with that, I'd like to first thank my colleagues, Ping Yan, who's the chemist who synthesized all those fluorescent voltage sensors, the voltage sensitive dyes, and Corey Acker, who's the neuroscientist and who did those beautiful experiments on the brain slices. And this is our company, as Bruce mentioned. And I'd like to thank all of my colleagues in CCAM. The top two rows show all of my faculty colleagues over the last 20 years or so. And almost all of them I've had either one or two papers with or had a grant with. So it's really been a very, very collaborative environment. And I'm very grateful to them for all of their multidisciplinary input to make all this work possible. And the third and fourth rows show some of the researchers, either graduate students or postdocs or research assistants or research associates. And in one case, I've even highlighted an undergraduate who has made tremendous impact in the last couple of years, a UConn undergraduate. And thank you, UConn, for finally giving us access to your great undergraduates here at the medical school. So, and then one more slide. Thank you to the family. This is the kind of occasion where I hope you'll indulge me and let me show off my family. It's another piece of work that I'm truly proud of. And it's a piece of work that I couldn't have done without my loving partner, Helen, who I think is in the audience. And with that, I'd like to open the floor to questions.
Well, Dr. Loew, that was really impressive. And even with my playwright self, I have a million questions. But we have about 10 minutes or so, maybe just a little bit more, a few minutes more than that. And I'd like, we have a really great audience here, over 40 folks. And so I'd like to certainly open it up to folks in the audience who would like to ask a question before I jump in with my playwriting silliness. And let's not stand on ceremony. Please just unmic and feel free to ask your questions. I'll give just a little bit of time for that to happen. And if not, I will certainly jump in with my silliness. So you all had better get on point. I see some hands coming up, and I can't see people, but please just unmute and ask your question if you don't mind.
Les, can I ask you a question?
Sure.
Can you tell us the more advanced voltage sensitive dye that your company is actually produced as a product, and what might be coming down the pipe as well, to the extent you are comfortable sharing? Just curious.
The best voltage sensitive dye? Okay, I'll tell you. It's di4ANEPQFPTEA. How does that help? So we're actually thinking about coining some new names for the dyes because people have a lot of trouble with the names that we use. So we're thinking about things like electrofluor or something like that as a new name for these dyes. But the best dye produces a voltage response that's about a 20% change in fluorescence in response to an action potential. And we're working on a new technology, and that's sort of about as good as it gets for voltage sensitive dyes. We're working on a new technology that should improve that by an order of magnitude, but we still have a lot of work to do on that.
Wow, appreciate that. Well, before anybody jumps in, I'll ask this question really quick. This is a quick and easy one. Dr. Loew, the one thing you talked about, a process you talked about earlier was collaboration. And even as a playwright, that's a space that I'm well familiar with our work in the theater. I wonder if you could take a minute and talk about like what was your most exciting collaboration? What was the work focused on? And you know, was it an outcome that rose to the occasion of the collaboration?
The virtual cell project has definitely been a collaboration that I'm probably the most proud of.
Mm-hmm.
It involved as I said, the full gamut of chemistry, physical chemistry, really, computer science, mathematics, physics, and of course biology. And that's probably the best example of this multidisciplinary approach that we use here.
Fantastic. I appreciate that. I won't get in the way. The floor is open. Please unmute and ask a question if you'd like. We still have plenty of time. Don't be shy. I will jump in and ask another question while we are waiting, Dr. Loew. Forgive my playwriting questions coming at you. We talked a little bit about, I think it was before we got together with the folks coming in the door, about the mysteries of the body. And as I listened to you talk, I thought, we know so much. We have this technology that gets us right down to the crux of what is happening in that particular cell or in that particular system. There's even some chaos going on that is also determinate. I'm curious what you think is or what you feel is a mystery that you are undone by, and how you think you might get at that particular mystery.
I mean, there's so many. I mean, it's certainly, if I gave the impression that we understand so much, that's not the impression I wanted to give. It's really that we understand so little. And I think that what excites me the most is that we understand a lot of the pieces. You know, what I spoke about were some of the fundamental physical chemical laws that drive all chemistry and all physics. And after all, the cell is just a very complicated chemical factory, or maybe you could say that it's a set of interacting chemical factories. So if we can understand that, we can understand how the cell is working. But there's so much that we don't know. There's so much data that we still need to be able to even build a model of a cell. People are attempting to do it, and that attempt is very laudable, but what we're trying to do is try to understand individual small systems in the cell, like that filtration barrier, for example, or like the dendritic spine that I mentioned. If we can begin to understand that at some level of detail in a predictive way, then we can take that information and connect it together, and then begin to build more and more complex models. And that's really what the philosophy is behind the virtual cell database, is that people can copy and paste pieces of one model and put it into another model, build more and more complex systems that can explain emergent properties that you couldn't possibly predict ahead of time, like the chaotic property of the kidney podocytes, the foot processes in the podocytes.
Fascinating, fascinating. Appreciate that. Once again, I'm sending out a call for questions from people who are way smarter than me, especially in this area. I see a hand, please, sir.
Dr. Loew, thanks for a very excellent talk. I had a small question about the dendritic spine that you presented a model of. Is the purpose of spine merely to increase the resolution of detection from signal outside? Or you say that the spines work independently, then how do signals get integrated and spatially remain segregated within a fiber?
Yeah, well, those are all very fundamental questions. The spine acts as the initial sort of amplifier of the incoming signal. And then that gets integrated by the dendrite. And how it's integrated depends on which other spines on that same dendrite might also be activated within a certain period of time of the activation of the first spine. So you can get integrated signals that travel from the dendrite down to the cell body, to the soma of the neuron, where if they're strong enough, they can set off an action potential that goes down the axon of the neuron. So, you know, exactly what the timing is and what the spatial relationship is between the individual spines that are activated is what controls all that. But the fundamental event is the amplification of the signal, or the compartmentalization of the signal in the dendritic spine.
Fantastic. Folks, we have a few minutes left. I know we can get another question in. We cannot let Dr. Loew go without getting everything we can out of him today. Board of Trustees, distinguished professor. So please, please unmute and jump right in. I know you are done hearing these silly questions from the playwright. We've got a couple of, is there anyone else?
I'll ask a question.
Please.
Okay, so, you know, your talk was excellent. It was a little bit over my head still, but I am wondering, you know, your students, what do they go on to become or do with their degree? And yeah, if you could just speak to some of what your students have done.
Well, I mentioned Sherry Ann Brown, who's now a physician scientist in Wisconsin. And I mentioned Sibeli Falkenberg, who actually had training as a mechanical engineer. That's what she did her PhD in. And then she came and did a postdoc with me, and now she's the head of a computational biology group at GlaxoSmithKline. Another student that was in that rogues gallery at the end, John Ditlev, did a joint project with myself and my colleague Bruce Mayer. And Bruce is an experimentalist, and he did some really excellent experiments, but then he modeled it with virtual cell under my supervision. And now he's got his own lab at the University of Toronto after doing a very good postdoc. So, I mean, some go into academia, some go into industry.
Fantastic, fantastic. Well, I thank you for that, Amanda. Well, everyone, I think that we are out of time. Dr. Loew, you have left us with that wonderful imagery of a rogues gallery. I think I'm going to steal that from time to time. I appreciate that. For all of those who joined us, we cannot thank you enough for taking the time to celebrate Dr. Loew and his Board of Trustees Distinguished Professor position. We thank you once again. We hope that you'll join us for the next conversation that we have, and I hope everybody has a wonderful evening.